Hi, folks. I'm a longtime Odyssey Symposium listener and first-time Odyssey Symposium speaker. So today I'm happy to talk about our work comparing probabilistic and rule-based phenotype algorithms for hypotension and angioedema to the experience observed in randomized clinical trials. Here are our disclosures. So rule-based phenotype algorithms are the standard for identifying outcomes in observational data. However, the performance characteristics of the phenotype algorithms, things such as sensitivity and positive predictive value, are often estimated to be low for many phenotypes. Probabilistic phenotype algorithms, those derived from logistic regression models, offer an alternative to the rule-based method. Prior efforts have demonstrated the potential for probabilistic phenotyping as an alternative to rule-based phenotype algorithms. The objective of this study was to develop a methodology for creating probabilistic phenotypes and to show examples of its use in hypotension, low blood pressure, and angioedema, a subcutaneous tissue swelling triggered by an allergic reaction. So here's something you all know. Here's an example of a rule-based phenotype for angioedema, where you might find a code, either an IC9, IC10 for angioedema, and that would be the cohort entry event, and then you set a cohort exit event. This is what a probabilistic phenotype might look like for angioedema, where you have a bunch of covariates that were selected by the regression model, all indicative of the condition, with a weighted beta coefficient. So for instance, you might have things like an angioedema code within there, which got a certain weight, allergic disposition, an emergency room visit. So for rule-based phenotype algorithms, the way you use them is you create the rule-based phenotype algorithm, and then you find subjects during the time at risk in your cohort of interest satisfying the algorithm. For the probabilistic phenotype, you use noisy labeled positive and negative controls to develop a supervised learning probabilistic model using lasso regularized regression. And by positive controls would be people with a high probability of having the condition of interest. Negative controls would be those with a low probability of having the condition of interest. We then applied our model at each appropriate time point during the time at risk for each subject in the cohort of interest. And from there, we selected the highest probability among the different cut points within the time at risk for each subject. Then we have to use a probability cut point, a point where anything above it would be considered somebody with the condition, anything below would be considered without the condition. To evaluate our models, we conducted our work in 5 administrative claims data sets. For the rule-based algorithm, we used a single occurrence of a diagnosis code for hypotension or angioedema. For the probabilistic phenotypes, we developed the probabilistic phenotypes and examined the results using different probability cut points from a range of 0.5 to 0.9. We then estimated the incidence of angioedema and hypotension while on treatment for 7 antihypertensive and 2 anti-diabetic drugs. Using both the rule-based and probabilistic phenotypes, we performed the analysis on 9 new drug user cohorts between 2010 and 2023. For our metrics, we extracted the incidence estimates in 95% confidence intervals from the results from the randomized clinical trials as a basis for comparison. We computed the proportion of incidence estimates for the rule-based and probabilistic algorithms that fell within the 95% confidence intervals for the incidence estimates from the clinical trials. We also assessed the performance characteristics, in this case positive predictive value and sensitivity, of the rule-based and probabilistic phenotypes using the Odyssey 2-Wolf evaluator. So this is some of the results for angioedema. On the left, you can see the 11 clinical studies that we used for 9 different drugs. The black squares indicate the incident estimate that was given from the clinical trials and the 95% confidence intervals. Here we have the results from the probabilistic and rule-based assessment. Those for the probabilistic are in stars. Those for the rule-based are in diamonds. Those that are in green fell within the 95% confidence interval. Those in red fell outside the 95% confidence interval. What we found was for the probabilistic algorithm at a 0.67 cut point that 77% of the estimates from the probabilistic algorithm fell within the 95% confidence interval, while we found for the rule-based 23% fell within the 95% confidence interval. For the performance characteristics, we also found better sensitivity and positive predictive value for the probabilistic over the rule-based algorithm. We found similar results for hypotension, again for a cut point of 0.67, where for the probabilistic algorithm we found a 78% fell within the 95% confidence interval, whereas 11% for the rule-based fell within the 95% confidence interval. For both angioedema and hypotension, the 0.67 cut point provided the closest match for the RCT results. Lower cut points produced higher average incidence estimates compared to RCT results, and higher cut points produced lower average incidence estimates compared to the RCT results. In conclusion, then, probabilistic phenotype algorithms for angioedema and hypotension estimated the incidence closer to the results from RCTs compared to those for the rule-based phenotype algorithms. The performance of probabilistic phenotype algorithms was superior to rule-based based on the characteristics of positive predictive value and sensitivity. Future research is still needed to evaluate the performance of probabilistic phenotype algorithms in post-market safety studies and determine how they could potentially be used to estimate the incidence of drug adverse effects. And thank you. And if you have any questions, please come to poster 115.